As promised, here's a recording. I was going to do a video, but unfortunately the file's too big to post. So just me speaking and talking about the slides. I think we went over this, but we're going to go ahead and go over it again because it's some important information. And I'm going to try to highlight the information that's going to be really important for the exam for you. So this slide is liver functions, and we know that the liver detoxifies as part of carbohydrate metabolism, lipid metabolism, protein synthesis, and secretion of bile. Talked about that in class on Wednesday. But a reminder that the detoxification process is due to the Kupffer cells in the liver that are phagocytes, and they phagocytose foreign substances. The liver also makes chemical alterations, changes hormones. It will change the drugs that we take to an excretable product, hence making it less toxic. Urea, uric acid, things like that make things less toxic, and allows for excretion of these types of molecules in the bile. Now, with carbohydrate metabolism, we remember that glucose is stored as glycogen way back from the beginning of the semester, and then also fat, and vice versa, right? Glycogen becomes glucose. So not only does the liver store glycogen and some fat, but can also break down the glycogen and glycogenolysis, as well as create new glucose molecules from proteins and fats via gluconeogenesis. And then secretes. The liver has the ability to then secrete the glucose into the bloodstream for use in other regions like skeletal muscle. Also, lipid metabolism. Now, this is the synthesis of triglycerides as well as cholesterol. The importance of this is we have cholesterol within our bile. The problem is the cholesterol can actually cause gallstones when it collects in the bile. Also, liver is important in the production of ketone bodies, in the metabolism of fat or lipids, and their production. And overproduction causes ketosis, and these are acidic. Now, protein synthesis is really important because it produces albumin, as we talked about earlier. And albumin, remember, is the main protein in the blood that really serves to keep oncotic pressure between interstitial space and the plasma, blood plasma. And then there's many other transport proteins, many that we've talked about. And the other important factor under protein synthesis is clotting factors. When someone goes through liver failure, one of the problems is they stop making clotting factors. And so we'll have hemorrhages much easier. And finally, of course, is the synthesis and secretion of bile. It synthesizes, conjugates it, and then secretes it, as we know, into the gallbladder. There's our Kupffer cells, there's albumin. So bile itself, about 250 to 1,500 mils a day. So that's a lot of bile. The pigment of bile is actually green, and in a cadaver, those of you that may have seen the front section, or the cadaver was lying supine, may have seen that the bile, or excuse me, the gallbladder itself is quite green. It's because of that bile pigment. It's bilirubin. Bilirubin is actually produced in the spleen, the bone marrow, and the liver. And it's a byproduct of red blood cell and the hemoglobin within the breakdown. Now, in bilirubin, when it's secreted, is converted to something called urobilinogen by the intestinal bacteria, and that's what makes our feces brown. And then bilirubin, if it's not excreted, can also return to the liver to be recycled. It also can be excreted in the urine. When we do the urine lab, we're actually going to test for urobilinogen in the urine. Okay, here's the metabolism of heme to bilirubin. So you have the red blood cells that go to the heme that break down, right, in the spleen, liver, bone marrow, and produce hemoglobin, which is then transformed into heme. And there's a removal of the iron at this point, thus recycling it in the bone marrow for recreation. Next, we have the heme turning into biliverdin. And biliverdin is just a step in the pathway. It's not hugely important, other than it being a step. But notice here that the NADPH, this is another proton carrier, and uses phosphates. But basically, it's going to donate its hydrogen to form the bilirubin. And in the liver, the bilirubin is conjugated. Okay, it's called bilirubin glucuronide, and conjugated bilirubin, it allows it to be excreted in the feces. So there's a little bit more detail on how it's excreted, and that it has to be conjugated in order for that excretion to take place. And then kind of looked at something similar to this picture on the right here. And basically, starting here with the liver, we have secretion of the bile, right? It's stored in the gallbladder, and then secretion into the bile, which contains bilirubin, into the small intestine, where the intestinal bacteria converts it to urobilinogen, and there we have urobilinogen in the feces. But also the urobilinogen enters the general circulation, and that's how it gets into the urine, because that general circulation then carries it to the kidneys, and urobilinogen is excreted in the urine. All right, bile acids. This is when there's a breakdown of cholesterol, basically. There's a whole breakdown pathway of cholesterol, and this is what forms bile salts. And the bile salts end up aggregating, and they aggregate as a micelle. And this is an example of a micelle, where the green portions here, that represents the non-polar portion. All of the non-polar areas are on the inside. And then this line on all the outside areas represent the polar, right? And so that allows the micelles to dissolve in water, right? It's because the entire outside is polar. Okay. And really all these bile acids are absorbed, they're reabsorbed, and that's what the ileum, a big part of the ileum, 95%. So that end portion of the small intestine is really important in that reabsorption of the bile acid. Now, for detoxification of blood, I've gone over most of this, but just a reminder, there's 3 ways. One is it's excreted into the bile. 2, it can be phagocytosed by the Kupffer cells. And 3, there's chemical alterations performed by the hepatocytes. So, for example, we get ammonia whenever amino acids are deaminated. And ammonia is very harmful to the body. So ammonia can be converted to urea, and urea then is excreted into the urine to rid the body of excess ammonia. All right, secretion of glucose, triglycerides, ketone bodies. The liver really is important with blood glucose level balancing. Examples, as stated before, glycogenesis, lipogenesis, both of those performed by the liver, and then, of course, the breakdown of glycogen into glucose, glycogenolysis. And then, as mentioned prior, the gluconeogenesis and the ketogenesis. All right, albumin and most plasma globulins, pretty much all of it produced by the liver. Remember that albumin makes up about 70% of the total plasma protein, and that's where we get the colloid osmotic pressure that we talked about in previous chapters. And then, of course, the clotting factors. And also the liver produces something called angiotensinogen, which is converted to angiotensin 1 and then to angiotensin 2, which is a vasoconstrictor. So therefore, it helps regulate blood pressure and blood volume when the blood volume is acting on the kidneys. We'll get into more detail about that when we talk about the kidneys. All right, the gallbladder. It's a sac-like organ that's green, and it stores the bile. It concentrates it. Whenever the small intestine is empty, the sphincter of Oddi, I always say Odie because it reminds me of Garfield and the dog, Odie. All right, you know me, I'm a little strange. But the sphincter of Odie in the common bile duct closes, and the bile ends up being forced into the gallbladder for storage because there's nothing in the small intestine at the time. And that gallbladder can expand, and it can expand pretty well into a pretty large compartment. But when food is in the small intestine, that's when the sphincter of Oddi opens. Gallbladder will end up contracting under the influence of cholecystokinin or CCK, and the bile is ejected through the cystic duct into the common bile duct and then into the duodenum. So here's a picture. There's those gallstones I was talking about. A lot of them are cholesterol based, and you can see this is an actual gallbladder showing the presence of gallstones. And I just look at that and I think, ouch. All right, let's talk about the pancreas. The pancreas has endocrine functions we've talked about in the endocrine portion, but let's talk about the exocrine functions. In this case, it's a different type of cell. It's called an acinar cell, and that's what makes the pancreatic juice. And the pancreatic juice is basically a bunch of enzymes with bicarbonate and other substances. And it's delivered to the duodenum via the pancreatic duct, and that duct kind of runs through the center of the pancreas. And you can see that here. There's the duct, there's the acinar cells here, and you can see the secretion of bicarbonate and inactive enzymes. Okay, let's talk about pancreatic juice. It's made up of that bicarbonate enzymes like I mentioned, but also water. There's enzymes in it for 3 classes of macromolecules: amylase for starch, trypsin for protein, lipase for fats. We also have brush border enzymes that are required for complete digestion, and this is because it's needed to activate trypsinogen to trypsin, and then trypsin then goes on to activate the rest of these enzymes that break down peptide bonds. So looking at this list, we have trypsin, we have chymotrypsin, we have elastase. Sorry, they're not perfect, but elastase and carboxypeptidase. All of these are going to break down peptide bonds. The first 3 cleave internal peptide bonds, and note that Trypsin is the activator for chymotrypsinogen, proelastase, and procarboxypeptidase, as well as phospholipase. But we haven't gotten into the fats yet. The only difference really between chymotrypsin elastase and carboxypeptidase is the internal versus external or terminal end of polypeptide. The carboxypeptidase really cleaves that terminal end of the polypeptide. So then we have the phospholipase, and that's an interesting one because it does cleave fatty acids from phospholipids. And so we're talking lipids, and it's a lipase, but it's actually activated by trypsin, so it's a unique one. Then we have plain old lipase. Doesn't need activation because it wouldn't break down the pancreas. The amylase, same thing, doesn't need activation. And then we have three others that are pretty unique. Cholesterol esterase. This releases cholesterol from its bonds with other molecules, so we get free cholesterol. And then ribonuclease and deoxyribonuclease. They cleave, based on their name, it's pretty easy to remember. They cleave RNA and DNA to form short chains of nucleic acids. Okay, so here's what I was talking about, this activation of the zymogens that come from the pancreas. So here's the brush border enzyme. And so we have enterokinase. And this is the important brush border enzyme because it takes trypsinogen and makes trypsin. And as you saw previously, trypsin is very important in activating other proteolytic enzymes. So this is an important slide. Hint. All right, bicarb. So the cells that actually line the ductules in the pancreas, some of them are acinar cells, but there's other cells that are just bicarbonate formation cells. And what it does is you take CO2 from the blood, or the cells take CO2 from the blood and form carbonic acid, which is H2CO3. And that will readily dissociate into hydrogen ions and bicarb. So the bicarb gets secreted into the pancreatic juice and the hydrogen goes back into the blood. And how the bicarb gets into the pancreatic juice is it's counter-transported. So Cl will enter the cell while the bicarb is secreted into the pancreatic duct. Note that people with cystic fibrosis really have trouble secreting bicarbonate because they have problems with chloride transport. These carriers are defective. And one of the problems not only is issues with mucus in the lungs, but mucus buildup within the pancreas, and the pancreas actually is destroyed. Here's a picture of what we just talked about. Really what's important on this slide is note that the carbonic acid dissociates into hydrogen, which is pumped out into the blood, and the bicarbonate, which is countertransported into the pancreatic lumen with chloride ion. And note that the chloride ion just passively diffuses into the lumen of the pancreatic duct. The CO2 passively enters again to form the carbonic acid. Don't worry too much about all this stuff right now. All right, let's look at neural and endocrine regulation. The nervous system and the endocrine system modify activity of the GI system. So remember, vagus nerve is primarily parasympathetic, so it's very heavily involved in regulating and coordinating digestive activities. So the GI tract ends up being both an endocrine gland as well as a target for hormones from other endocrine glands. So for example, the stomach secretes gastrin, and that stimulates parietal cells to secrete HCl. It stimulates chief cells to secrete pepsinogen, and also helps maintain structure of the gastric mucosa, the bicarbonate-rich mucus. And the small intestine will secrete secretin, and note that that stimulates both water and bicarb secretion in the pancreatic juice, as well as helps potentiates the action of cholecystokinin. Because cholecystokinin, when it works on the pancreas, that's the stimulus for pancreatic juice. So they work together. The small intestine, cholecystokinin, big player. It stimulates contraction of the gallbladder, stimulates secretion of the pancreatic juice enzyme, but it also inhibits gastric motility and secretion in the stomach, right? The food has moved into the small intestine. Don't need the action of the stomach anymore, so CCK inhibits that motility, as well as secretion of HCl. And it also, just a side thing that it does, is this maintenance of the acini in the exocrine pancreas. But really, these three actions are important actions to know when it comes to CCK. All right, and then GIP, or gastric inhibitory peptide, or glucose-dependent insulinotropic peptide. I like just gastric inhibitory peptide. This actually also acts on the stomach, so it inhibits the gastric motility and secretion, but it also stimulates secretion of insulin, because at this point you're in the fed state. We'll talk more about fasting and fed state, but you've just eaten, the food has entered the small intestine, so insulin is going to be needed for all of the glucose molecules that will be absorbed to enter cells. All right, gastric motility and secretion occur automatically, right, via the enteric nervous system. The waves that we talk about when we talk about peristalsis, they're actually initiated spontaneously by cells called pacemaker cells, or pace setter cells, excuse me. And secretion does not even need hormone or neural inputs. Now, it's important to note that the enteric nervous system, the autonomic nervous system, and the hormones affect, superimpose or act on top of that automatic activity. So you have not only this automatic enteric nervous system that's separate from everything, but you also have the overall autonomic nervous system and overall endocrine system that assists with all of these activities. Now, extrinsic control of gastric function ends up being divided into three phases that are important to know: cephalic, gastric, and intestinal. So cephalic is triggered by sight, smell, and taste of food. And the control is via the brain, via the vagus nerve, and it stimulates the ECLs, the chief cells, and the parietal cells, basically the cells in the stomach. And they start secreting in the thought that food will be entering the stomach very soon. It lasts for about 30 minutes. I think it begins around 15 minutes prior, and then for the first 30 minutes, there's this cephalic phase stimulation during a meal. Now, the gastric phase is triggered by the actual arrival of food into the stomach. And this is where the secretions are stimulated by stomach distension, and as well as what is in the bolus that enters the stomach. So positive feedback will occur, and it's usually proteins. And so think about it as more proteins that are broken down, the more secretions are released to break them down. Right? Glucose has absolutely no effects, and fat actually inhibits gastric secretion. Say that again. Fat inhibits gastric secretion. Now, not only is there positive feedback, but there's a negative feedback. As the pH drops, a lot of times because a lot of HCl is being produced, and the pH goes down to less than 2.5, the hormone somatostatin is released. And this will inhibit the gastric secretion and really will stop it at a pH of 1, so it doesn't go below that. Note that large amounts of proteins and polypeptides will sometimes buffer the pH, so the secretion has to match the protein concentration. That's kind of an interesting fact. And here's the regulation of gastric secretion. You have the vagus nerve. We have gastrin. Okay. Gastrin secreted by the G cells that acts on the ECL cells. Okay. Histamine from the ECL cells act on the parietal cells. So you have gastrin stimulating the ECL cells to then release histamine to act on the parietal cells to secrete the HCl into the stomach lumen. Okay. So note that the G cells, that stimulation of gastrin, can be enhanced by not only the vagus nerve, but amino acids, which make up protein. Right, the intestinal phase begins when the chyme enters the small intestine, and of course there's inhibition at that point of gastric activity. The arrival of chyme is detected by the neurons of the vagus nerve, and that reflex is a neural reflex, again, that inhibits motility and secretion. So I mentioned in the previous slide the fat in chyme has a role both in the stomach and in the small intestine, but mainly in the stomach. So the fat that's in the stomach and in the chyme that enters the small intestine will stimulate the small intestine to secrete enterogastrones. And these are hormones that inhibit motility and secretion in the stomach. So they include what we talked about earlier, somatostatin, but they include CCK, GIP, and GLP-1. And remember, GIP and GLP-1, when we were talking about some of these hormones, they stimulate insulin secretion, and it's just done in anticipation of glucose entering the blood from digestion. All right, some intestinal reflexes. There's 3 of them. Let's talk about them. They're mediated by the enteric nervous system, paracrine hormones, and then, of course, regulated by the autonomic nervous system and hormones. So the gastroileal reflex refers to increased motility of the ileum. And remember, that's the 3rd portion of the small intestine, and therefore increases and stimulates the movement of the chyme through the ileocecal sphincter in response to increased gastric activity. So it's an interesting thing that the stomach affects the ileum and pushing the food through to empty the small intestine in readiness for the next round of food. The ileogastric reflex decreases gastric motility in response to distension of the ileum. So again, we have, instead of emptying the small intestine or the ileum into the cecum, which is part of the large intestine, in this scenario we have a decrease of the gastric motility when the ileum is distended. So, and then the final one, if any part of the intestine is over distended, it's going to cause relaxation of the smooth muscle. So that's called the intestinal reflex. So anytime there's that distension in one part, it will cause just relaxation through the entire small intestine. Okay, secretion of pancreatic juice. And we've talked about a lot of this. I like to repeat things, state them in different ways on the slides so to enhance your learning. But remember that secretion of pancreatic juice is stimulated by both secretin and CCK. But let's go into a little bit more detail. Secretin is secreted in response to low pH in the duodenum. And so immediately upon entering the duodenum from the stomach, the pH of the chyme is going to be quite low. And therefore, secretin is there to immediately cause release of bicarb from the pancreas into the small intestine to buffer that acidic chyme. And then CCK is secreted in response to fat and protein content because it stimulates production of the pancreatic enzymes. It also enhances secretin, so that means more bicarb, and stimulates contraction of the gallbladder so that fat is broken down in or emulsified into smaller fat droplets for digestion. Okay, let's quickly go into detail about some of these enzymes and how they do their job. So remember, salivary amylase begins starch digestion in the mouth. And then we have pancreatic amylase, and it converts starch to oligosaccharides, but also to trisaccharides and disaccharides. So here we have an example of oligosaccharide, right? A trisaccharide and a disaccharide. And so the oligosaccharides, because they're still fairly large, have to be hydrolyzed by brush border enzymes into maltose and then maltose. And of course, finally, into glucose. In digestion, absorption of protein, remember it begins in the stomach with pepsin, and they form polypeptides. And then in the small intestine, we have the pancreatic juice enzymes, trypsin, chymotrypsin, etc., that cleave peptide bonds. And the result ends up being free amino acids, dipeptides, tripeptides. Now, free amino acids are absorbed by cotransport with sodium into the intestinal epithelial cells and then immediately secreted into the blood capillaries. Di and tripeptides are carried into the epithelial cells, and within the cells, they're hydrolyzed into those free amino acids and then secreted. So this is just a picture of what we just went over. There's the dipeptides. There's dipeptides and tripeptides entering. As you can see, here's a tripeptide, and the tripeptidase is located within the epithelial cell. Breaking it down. Okay, digestion and absorption of lipids is a little bit more complicated. This, of course, occurs in the small intestine, and anytime you have arrival of lipids into the small intestine, we have secretion of bile from either the gallbladder or directly from the liver if someone lacks a gallbladder. Fat is then emulsified by these bile salts. The point is to increase the surface area for fat digestion. They form tiny droplets of fat. I showed you earlier what a micelle looked like. This one's an even better representation because this is fat absorption in the small intestine. That you can see the bile salts here on the outside in yellow, and then the glycerol, and then the actual fatty acids all pointing into the middle of the micelle. Okay, this is just showing you an example of what occurs in the small intestine. So you have the bile in green. It's forming a micelle, bile salts, cholesterol, lecithin. And you have the fat droplets. So then those become emulsified fat droplets. You add in lipase, you get free fatty acids and monoglyceride that then form into the micelles. And then we have digestion. So pancreatic lipase hydrolyzes the triglycerides by adding water to free fatty acids and monoglycerides. The absorption occurs of these free fatty acid monoglycerides um, when they are absorbed into the epithelial cells. Inside the epithelial cells, they're resynthesized into triglycerides and phospholipids. So it's an interesting point that the triglycerides and phospholipids are broken down in the small intestine in order to get into the epithelial cells, where they are resynthesized into their triglycerides and phospholipids for entrance into the lacteals. Remember, this is the um, lymphatic system here, and eventually ends up in the thoracic duct. All right. Now, before they enter the lacteal, the triglycerides and the phospholipids will combine with a protein. And that particle, that combination of protein, phospholipids, or triglycerides, is called a chylomicron. And again, secreted into the central lacteals of the villi. Transport of lipids is done when the chylomicrons combine with apolipoproteins. And this allows them to bind to receptors on capillaries in both muscle and fat. And endothelial lipoprotein lipase will hydrolyze the triglycerides into the free fatty acids and glycerol. And therefore, this provides energy for muscle, also storage in adipose tissue. And then any cholesterol containing remnants are then taken up by the liver. Okay, transport of lipids continued. These are really interesting to me. They should probably be interesting to most of you because this is cholesterol. This is a cholesterol test, and this is what you're going to see on if anyone has ever had their cholesterol checked. So it's part of transport. And so the cholesterol and triglycerides from the liver will form VLDLs, and that's very low density lipoproteins. And they get secreted from the liver, and their job is to take triglycerides to the cells. So once at the cells, those triglycerides are removed and the VLDLs become LDLs, low density lipoproteins. And these transport cholesterol to the organs and blood vessels. So we call this the quote unquote bad cholesterol. So you do not want high levels of LDLs. Now, HDLs will transport excess cholesterol back to the liver. So we pretty much like this stuff, right? We don't want too much cholesterol floating around in our bloodstream. So a high ratio of HDL cholesterol to total cholesterol, we believe confers protection against all that gumminess, the atherosclerosis that occurs in blood vessels. So that's where it gets that good cholesterol name. And that's the end of digestion. I'm posting this. I hope it helps you study. I will go over any slide that anyone wants me to go over on Monday. If for some reason class is canceled on Monday because of air quality, then you'll have this to finish digestion. I will also post the next chapter as well, and that way we're covered for the exam on Wednesday. And I believe it should be raining by then, and our air quality should be better. I hope. Cross those fingers. Have a great weekend.